Hello, people. This is the Quiz 6 review. I messed up and I forgot to hit record on Zoom till about halfway through. And a bunch of people's faces were in there anyway. So I'm just going to go ahead and re-record this whole thing. It's going to go kind of fast, but you can slow it down if you need to, skip around, or go back. So, without further ado, methylation. This is the start of our epigenetics. Methyl groups, which is CH3, are small molecules that are added to C's that are next to G's, CG islands. If you put a methyl group on a C, that is going to chemically change it ever so slightly. And if you do that to enough C's, you are actually going to make it difficult for the cell machinery to kind of work around all of this methylation. You can think of the methyls as kind of little spiky things on the C's that kind of interfere with the normal processes. So, if you methylate something, you're normally going to get lower expression of that gene. Methylation, lower expression, less proteins. You can determine if genes are methylated with a chemical test called bisulfite treatment. Bisulfite normally turns C's into U's. U's get changed into T's when you do PCR, so you can think of it as C's get changed to T's. But this only happens when they are unmethylated. So an exposed C, a non-methylated C, will get changed to a T. But a methylated C has a little hat on it that blocks the bisulfite. So the C will stay a C. So you can get a control, which is an untreated sample. Just has the C's as they normally are. And a treated sample, where you're going to see some C's and T's and whatever. If there are any C's that ended up changing into T's, you know that they are not methylated because they got hit. If they stay the same, they are methylated. So when you count methylated versus non-methylated, remember, if they change, they're not methylated. And if they're methylated, they do not change. So it's kind of an inverse relationship there. Okay. Usually we're looking at methylation in a promoter region. Anywhere can be methylated. That's usually bad for transcription. But if we say a promoter is methylated, we're probably just saying that gene is turned off. That's how it works. The polymerase cannot easily recruit to a heavily methylated promoter for the reasons I described before. Bunch of little spiky things. The polymerase doesn't want to go there anymore because there's a bunch of methyls in the way. So, methylation in a promoter turns off the gene. We did talk a little about acetylation. Acetylation opens up a gene and allows it to be transcribed. It creates physical space between a histone with DNA on it. So this space in between, plenty of room for a polymerase to get in. But if you have a histone over here and a histone over here, because there's no acetyl making any space, that's deacetylated, no acetylation, that's pretty closed off, right? Where is a polymerase going to go? There's no room. So, less transcription. Let's talk about a special form of methylation. Remember, methylation is turning off a gene. Imprinting is a specific type of methylating. Imprinting is heritable. There are a set of human genes, or a set of genes in the human genome that are imprinted, which means either the mother or the father turns off the copies that they pass down. So, if one of these two parents passes down a turned off copy, a methylated and imprinted one, and the other parent passes on one that's not imprinted, not methylated, so turned on, you are going to get one imprinted, one turned off copy, and one non-imprinted, one turned on copy. So every person in their genotype is going to have one imprinted and one non-imprinted version of each of these genes, regardless of sex. So what you have is 1 and 1. What you pass on, however, is going to be all or none, because this is sex dependent. When you go and make sperm or egg, If this is paternally imprinted, i.e. imprinted in the father, all sperm are going to get imprinted. And that means the eggs, none of them are going to get imprinted. If this is maternally imprinted in the mother's genome, that means all the eggs are going to get imprinted and none of the sperm will. So, if you get one sperm and one egg, that means you're going to go back, 1-to-1. That means you're going to get one copy that's turned off, right? That one doesn't matter. Who cares? You're going to get one copy that is turned on. That's the one that matters. The copy that's turned on is the determination of phenotype. And yes, an imprinted gene is inactivated because it's methylated. Here's the process for it. Gametes are made. You start out with 1 and 1, right? One's imprinted, one's not. Now when you make gametes, none of them are going to be imprinted in both sexes. Erasure occurs. And then, in one of the genders, printing is going to occur. Reprogramming. So if this is maternally imprinted, the mother is going to imprint all of her eggs, and the father's not going to imprint any of his sperm. Or vice versa, if it's paternally imprinted, as is this example here. A male's making sperm. If it's paternally imprinted, all the sperm are going to get imprinted. If it's maternally imprinted, right, none of the sperm are going to get imprinted. Definitely good to know how to do some pedigrees for this as well. A few activities for that exist. Now, the last form of epigenetics we discussed was X inactivation. Only one copy of the X chromosome is active in any given cell. If your genotype is XY, then great. Your genotype, well, you're going to have that one X active in all of your cells. Sorry. Oh, there we go. Oh, okay. Can't write. Okay, anyway. Organisms with more than one X chromosome, so XX females, or XXY males, or there's also triple X females, they're going to get one or, well, multiple maybe, turned off. They can only have one X chromosome turned on in each cell. Well, how do we get the calico cats? So this is totally random. Some of the cells are going to turn off one of the X chromosomes. Some of the cells are going to turn off the other X chromosome. So in this cat here, this area came from a bunch of cells where the orange copy was turned on, and the other, like this black area, came from a patch of cells where the orange one was actually turned off, and the black one is turned on. So, very interesting. How does this happen? There is the Xist chromosome. The Xist chromosome is either on or off. How is it off? If it is turned off, it is methylated. Methylation turns things off. If this is methylated, it is turned off and nothing happens. This X chromosome stays on. This is methylated, which turns this off, which keeps this whole thing on. If Xist is on, the thing is off. If Xist is off, because it's methylated, the whole chromosome is on. However, if Xist is on, it's not methylated, so it's on, it is going to make stuff. It is going to express this Xist RNA. The Xist RNA is going to coat the X chromosome. That is going to inactivate this X chromosome and turn it into a bar body. So, Xist is on, so the whole chromosome is off. Let's talk about biotechnology. We have two types of DNA in bacteria. We have chromosomal DNA, and we have plasmids. Chromosomal, that has all its essential stuff. Who cares? Plasmids, these are interesting. They're little circle pieces of DNA. They usually give the bacteria some sort of like supplemental abilities, like resistance to drugs or toxic properties. So I like to think of them as Batman's gadgets. They give this bacteria extra capabilities that it wouldn't normally have. right? Batman can live without his gadgets, because he's got, you know, his normal stuff here. But the gadgets help. They do things. Bacteria can actually give and take and trade plasmids and just pick them up from their environment and stuff. That's a property we can exploit, because that means we can hack the plasmid, and therefore the bacteria will pick it up, and the bacteria will do the stuff that we want. How do we do this? Well, first, we have to put in a gene that we want into one of these plasmids. So we have to cut out the gene with a thing called a restriction enzyme. It recognizes a very specific sequence of DNA, makes a cut in a specific place on both strands, so as you can see, it's kind of just mirrored from each other. And that is going to leave these sticky ends. That means these sticky ends can actually, like, reattach to each other. So if I cut a gene out with this enzyme, and I cut an area on a plasmid with the same enzyme, they're going to have corresponding sticky ends, which means I can stick this thing from the gene into another sticky end of the plasmid, and now they go together. Awesome! It's like a little puzzle piece. Sometimes the plasmid will just go back together. That's okay, that happens. So the plasmid won't always get the gene, but the gene will make it into some of the plasmids. So make sure you cut your plasmid and your gene with the same enzyme. Do not slice the middle of your gene, because you don't want stuff getting in the middle of your gene. That's bad. Now, there are other things you don't cut. You don't cut the ORI, the origin of replication, because this is needed for the bacteria to reproduce this plasmid. You don't cut the antibiotic resistance gene. You just don't. We'll explain why. Don't worry about this. You will cut this, but not important right now. So, now, you have stuck your gene in this plasmid, ideally right here, the middle of this LAC-Z gene. Usually you're going to have a bunch of enzyme sites called the MCS, the multiple cloning site. You're going to stick your gene there, and then you're going to take this whole plasmid and you're going to put it into bacteria. Bacteria will be chemically competent, so you treat them with a chemical that makes their cell membranes open up, and then they can pick up these plasmids more. They'll just pick them up. But not all the bacteria that will pick up the plasmid. Now, how do we make sure we only have bacteria that have our plasmid? We exploit this antibiotic resistance marker. This gives them resistance to a drug called, well, it can be any antibiotic, but we're going to say ampicillin, so AMPAR, AMP resistance. So that means if we put all the bacteria on a plate containing ampicillin, the ones that have this antibiotic resistance marker, because they have the plasmid, are going to live. The ones that do not have this antibiotic resistance marker, because they don't have the plasmid, are going to die. So, there you have it. Now, all the bacteria that have on your plate, they all have the antibiotic resistance marker. So, that means they live. They all have the plasmid. Great. So, don't cut this because we need it for this step. Now we have a plate full of bacteria that have our plasmid. How do we actually know that the plasmid contains the gene? As I said, the plasmid can close back on itself. So how do we know? Okay, A, it has the plasmid, right, because it's alive. It wouldn't be on this plate if it didn't have the plasmid. But that plasmid does not necessarily contain the gene of interest. That is why we have this LAC-Z gene on here. If LAC-Z is together, it's intact, it's going to make a protein that combines with XGAL. XGAL plus LAC-Z protein is going to make a blue pigment, and you're going to see these blue dots. That means they do not have our gene. However, what if we put our DNA in the middle of LAC-Z? Now LAC-Z is interrupted because it has something in the middle. It cannot make blue anymore, so it's just going to be white. So white colonies have our gene of interest. Go for the boring ones, not the fun blue ones. The boring ones. These ones have our gene of interest. Now, great. We can take this colony, we can mash it up, grab its DNA, do all sorts of fun stuff with it. We have that DNA forever and ever and ever. What do we do with it? Well, first, we have an example. Okay. Do we want to use EcoRV? No. Slices our gene. Do we want to use PSTI? No. Slices our AMP. Great. So, what's the only thing we can cut this side with? BAM. BAM is in the middle of our LAC-Z, so that's great. We make an end here. We stick that there. Now what about the other end? Do we use EcoPI? Ooh, close to our gene, but there's nowhere for it to go. That's unfortunate. Cell 1, though, goes right here. So our gene can get stuck right there. That interrupts the laxity gene, so this bacteria is going to be what color? White. And are the bacteria going to live? Yes, they have ampicillin resistance, AMPR. So, great. We have a plasmid. We put our gene of interest on it. Woohoo. Okay, anyway. We can do things with that DNA now. We can add it into another organism. That's gene addition. So, that gene that you have, add it in. Now that organism can express that gene. Maybe. You can take a gene that it already has and replace it with something else. That's gene replacement. Gene knock-in is kind of a specific type of replacement. We're saying, okay, let's say we have a sheep insulin gene and we want to give it mouse insulin instead. So we're doing a replacement, but it's the same type of gene, just a different version of it. Gene replacement is more broad, so I could replace a metabolic gene with an insulin gene, or like some sort of like brain gene with a pancreas. I don't know. Like, you go wild. You can imagine the implications. You can also knock out, so you can swap in a bad copy of a gene, like a recessive allele, and now you can break a gene to study what happens to the organism when it does not have the proper gene. Oh yeah, we say transgenic organisms are organisms with two or more sources of DNA. But yeah, we have our techniques. Great. We also learned about one more big concept. We have promoters. So, I actually like to start this one over here. Okay. How do we have a gene of interest. Woohoo. We have a promoter, let's say, right here. Sometimes we want to cut out a promoter sequence and get it in the plasmid as well. So we're going to have to cut, and cut. Now, we need to stick together the promoter and our gene of interest. We're going to use the same restriction enzymes so that they correspond and they can stick together. Awesome. Now you have a promoter and you have a gene of interest that's stuck together. Now you get this whole entire thing into a plasmid. So this has to have a matching sticky end with a plasmid, and so does this end. Now it's in a plasmid. Awesome. Now, you take this whole entire gene construct from your plasmid and put it into a mammalian embryo, like a mice embryo. That is going to make this promoter and gene in every cell of the body. They all have it. But that promoter is not necessarily on in every type of cell. The promoter can be expressed in different types of cell tissues. So let's say this is a pancreatic promoter. Then this gene is only going to get transcribed in pancreas cells. It's not going to get expressed in every cell of the body usually. So definitely be vigilant there. Where's your promoter expressed? It makes it into every cell of the body, right? It's there, it's present. It just doesn't actually turn on this gene until it's in the right type of cell tissue. Thank you for watching.